Hello, thank you for taking an interest in my poster. My name is Katie West and I'm a second year PhD student at the University of York. I'm interested in long non-coding RNAs in the immune system. In particular, I study CD4 T cells. These are really interesting because when T cells are stimulated, thousands of genes change during the T cell differentiation process. This makes them a really great model for studying long non-coding RNA function. We've previously shown that the long non-coding RNA MALAT1 is really important for T helper cells. A summary of this is shown in box 1. We found that in the early stages of T cell activation, MALAT1 is rapidly suppressed. But complete deletion of MALAT1 results in lower expression of the anti-inflammatory cytokine IL-10. We went on to show that MALAT1 regulates IL-10 by the key transcription factor MAF. Not only this, the MALAT1 knockout cells have enhanced immunity in different infection models because of this altered IL-10 expression. If you'd like to know more about this story, I've linked to our recent paper at the bottom of the poster. But because we know MALAT1 is so important in T cells, we wanted to further understand how this is happening. So we sent naive Th1 and Th2 cells from wild-type and MALAT1 knockout cells for sequencing. As shown in box 2, we found quite a striking phenotype. MALAT1 knockout T cells fail to up or down-regulate genes to the same extent as wild-type cells during T cell differentiation. So this suggests that MALAT1 knockout T cells have a differentiation defect. Because of this, we believe that the rapid suppression of MALAT1 in the early stages of T cell activation is to act as a brake. And this would ensure that aberrant T cell differentiation does not take place. To investigate the mechanism behind this, we then looked at the MALAT1 protein interactome using a process known as RAPMS. The RAPMS protocol is highlighted in box 3. Essentially, it involves creating a covalent cross-linkage between RNA and protein by exposing cells to UV light, lysing the cells and pulling down MALAT1 using long antisense biotinylated probes and streptavidin beads. There's then various stringent denaturing washing steps, and the samples are then sent for mass spec. As you can see from the enrichment graph, pull down of MALAT1 was very successful. Because of this, we sent the samples for a mass spec. Mass spec identified 150 proteins, 30 of which were significantly enriched in the crosslink samples, and these are highlighted in red on the graph. There was also another 12 or so proteins that were borderline significant, but also plausible MALAT1 interacting proteins. These 30 significant RNA binding proteins are listed in box 4, and these range from things like HNRMPs to SR proteins, which are all plausible MALAT1 interaction partners. Further examination of this list, we found that 29 of the 30 proteins are known RNA binding proteins, as they are found in RBP base. Not only this, 20 of them have previously been shown to bind to MALAT1. So this really gives us confidence that the proteins we're pulling down are in fact true MALAT1 RNA binding proteins. To follow up on this, as shown in box 5, we next examined the log-fold change of these RNA binding proteins during wild-type T cell differentiation. A handful of these do change and are significantly changed, but the log-fold change is quite modest. So, one method of narrowing down this list is to compare the significantly enriched RNA binding proteins with genes that are regulated by MALAT1 in T cells. This is because RNA binding proteins often regulate their own mRNA. This identified 3 proteins in the overlap, which are intricately linked together in a network. Further examination of these proteins, shown in the heat map, we find that when MALAT1 is knocked out, these RNA binding proteins are downregulated. Now, this is quite interesting because dysregulation of these RNA binding proteins have been linked to T-cell differentiation defects. So, we believe that the ability of MALAT1 to interact with these RNA binding proteins is a critical determinant of T-cell differentiation. And ultimately, this will have implications in mammalian immunity. So, to summarise, MALAT1 is rapidly downregulated in the early stages of T-cell activation, but its complete deletion results in a T-cell differentiation defect. We see this reflected in the lower expression of IL-10 and MAF. We've gone on to characterise the MALAT1 protein interactome in EL4 cells and believe the ability of MALAT1 to interact with RNA binding proteins is a key determinant of T-cell differentiation. To follow up on this, I'm going to be carrying out RAPMS in primary T cells, validating these RNA binding protein interactions, and following up with knockdown studies. So, thank you very much for coming to visit my poster. If you've got any questions, I'd really love to hear from you. Thank you!